to talk about why we, the challenges, because we don't have a good assay for wild-type Huntington. Doug McDonald may want to comment on that. So if you're in the audience, Doug, or anyone who's working in this. Okay, so I'm delighted to introduce the next speaker from Wave Life Sciences, who's the medical director. Danlin Xu is going to tell you about an update on the Wave program. Thank you, Danlin.
So first of all, we would like to thank the EHDN for inviting us to present our allele-selective approach to treat Huntington disease. So our company, Wave Life Sciences, is a genetic medicines company. We focus on developing oligonucleotide drugs to treat severe and rare genetic diseases. So this is our forward-looking statement. So today, I'm going to talk about WVE-003, that is our new class of oligonucleotide that is designed to treat Huntington disease. I will show you our mouse data of WVE-003, and then I'll talk about our first in-human study of WVE-003. And then lastly, I would like to provide a brief introduction of our newly deployed assay to quantify wild-type Huntington protein in CSF. So since wild-type Huntington protein has been shown to play important roles in neuron survival and functions, so we believe preserving wild-type Huntington protein will be beneficial to Huntington disease patients. Therefore, our approach to treat Huntington disease is allele-selective. That means WVE-003 is designed to lower mutant Huntington mRNA, but not wild-type Huntington mRNA. The allele selectivity of WVE-003 is achieved by targeting a variant of a single nucleotide polymorphism called SNP3 that is present on mutant Huntington mRNA, but not on wild-type Huntington mRNA. WVE-003 belongs to our new class of oligonucleotides that contains of our novel PN linkage bonds. We have found in our preclinical studies that incorporation of our PN bonds has improved drug properties of our oligonucleotides, such as tissue distribution, potency, and the durability of their effects. So allele selectivity of WVE-003 was evaluated in Hu97-18 mice using WVE-003 precursor oligonucleotide that has exactly the same sequence as WVE-003. So in these two, Hu97-18 mice are transgenic mice. They express both human mutant and wild-type Huntington transgenes. And the human mutant Huntington transgenes are SNP3 positive. So this is a mouse model we can use to evaluate allele selectivity of our oligonucleotides targeting SNP3. So in these two figures, orange bars represent mutant Huntington RNA expression levels, and blue bars represent wild-type Huntington RNA expression levels. So both human mutant and wild-type Huntington RNA expression levels were evaluated at 3 time points after the mice were treated, week 4, 8, and 12. So if you look on the right side of the column, WVE-003 precursor molecule lowered mutant Huntington RNA transcript, but basically spared wild-type Huntington RNA transcript. On the other hand, our pan-silencing control, as shown for the middle columns, lowered both mutant and wild-type Huntington transcript. So this experiment demonstrates that allele selectivity can be achieved by targeting a single nucleotide polymorphism, and also support that WVE-003 can be allele selective in vivo. So this is the study results of WVE-003 in BAC-HD mice. BAC-HD mice are also transgenic mice. but they express only human mutant Huntington transgenes. So some of their human mutant Huntington transgenes are SNP3 positive. So this is a mouse model we can use to evaluate the WAVO3 effect on lowering mutant Huntington. So in these two figures, the dots represent human mutant Huntington RNA expression levels. We also evaluate human mutant Huntington RNA expression levels at 3 time points after the mice were treated, week 2, 4, 12. So if you look on the right side of the dots, WAVO3 lowered mutant Huntington RNA expression level between 50 to 75%, and its effect lasted up to 12 weeks. And our pan-silencing control, as shown in the, for the dots in the middle of the figures, also lowered mutant Huntington RNA transcript, but its effect in this experiment is not as durable as WAVO3, especially in striatum. So with our preclinical data, we have initiated our first in-human study of WAVO3 in Huntington disease patients. The study is a dose-finding and also a proof-concept study, and we named the study SELECT-HD. The primary objective of the study is safety and the tolerability of WAVO3, but we will also evaluate WAVO3 effects on relevant Huntington disease biomarkers and the clinical endpoints. The study is double-blind, placebo-controlled, and the study consists of 2 treatment periods, single ascending dose and a multiple ascending dose. We designed the study to be adaptive. That means after we select the initial dose based on our PK/PD modeling using our mouse and monkey data, the later dose level and intervals of WAVO3 will be determined based on safety and PK of our prior completed cohorts and guided by independent safety monitoring committee. As you may recall, we discontinued 2 Huntington disease trials early last year, Precision 1 and Precision 2 targeting SNP1 and SNP2. The reason we discontinued the 2 studies are that we found the study drug concentration in CSF are low, and also we did not observe the mutant Huntington lowering. So in this figure, we found we show the preliminary results of WAVO3 in CSF. As you can, in comparison with our 2 study drugs, as you can see, WAVO3 showed improved PK profiles with higher concentration and longer half-life. So with our PK/PD modeling, we are confident, we are optimistic that WAVO3 can be in pharmacological range in lower mutant Huntington. So to support our allele selective evaluation, we recently deployed a new assay to quantify wild-type Huntington protein in CSF. So for this challenge, we first improved the existing assay to quantify total Huntington protein in CSF. Then we developed a 3-step procedure to measure wild-type Huntington protein in CSF. So the main concept of the assay is to use mutant Huntington antibody conjugated magnetic beads to deplete the mutant Huntington protein from the CSF, and then use our improved total Huntington assay to quantify wild-type Huntington protein in CSF. The final result reflects the wild-type Huntington protein level in CSF. So in summary, we are committed to a allele selective approach to treat Huntington disease. WAVO3 is our new class of oligonucleotide that contains our novel PN bonds and has been shown to have improved drug properties in our preclinical studies. In our ongoing SELECT-HD study, our preliminary data has shown that WAVO3 has improved PK profile in CSF with higher concentration and longer half-life. And based on our PK/PD modeling, we are confident that WAVO3 can be in the pharmacologic range in lower mutant Huntington. So our study is currently ongoing, enrolling patients, and we continue to activate the study sites. So finally, we would like to thank all the Huntington disease patients and their families in their support and participation in our studies. We'd also like to thank our scientific advisors and collaborators, CHDI, and companies we have worked with in our efforts to develop oligonucleotide drugs to treat Huntington disease. Thank you for your attention.
Thank you, Danlin. Perfect timing as well. So time for a question. Any questions for Danlin? Ed in the front row.
Sorry. This time.
Oh.
Sorry, Ed, I think my hand went up first. I was just wondering...
I'm sorry, Noina.
That's all right. Thank you very much.
It's difficult because of the lights. It's difficult to see people, so they have to go like this. Sorry,
Thank you
Noina.
very much for a lovely talk. I was just wondering about the coverage of WV3 in the HD population, because I think the strategy for 1 and 2 was that it would have better coverage, and that's why you had 2 different products. So can you tell us a little bit about the coverage over the HD population with your drug?
Per publication, SNP3 prevalence is around 40%, and we are actively screening patients to see whether our number will match publication.
And the ethnic mix?
I think right now most patients are Caucasian. So far, the patient we have screened.
Noina, sorry. Actually, when the questions come up, if the organisers could put the lights up, it's much easier, because otherwise I just see a big blur. Ed, you need to keep your question for the panel discussion. So